In this session, we will learn a little bit about working with the Ncbi Resources, beginning with their gene section as well as RefSeq. The NCBI, or the National Center for Biotechnology Information, is a part of the National Library of Medicine, a division of the NIH. The strengths of the NCBI are that it is a very comprehensive one-stop shop. The content, however, is very technically focused and not oriented towards the general public. A key part of the NCBI is the gene section, and within the gene section is a part referred to as RefSeq. RNA RefSeqs refer to the complementary DNA sequence and therefore have no introns. Each of these entries begins with the two letters NM. These are considered to be the normal version of a gene. Within those, you will also see the latest version. Sometimes there are updates to the content. Now let's go to the NCBI website, and then we'll choose from the drop-down menu to go to the gene area. After picking that, then we enter CFTR. When you do that, you can see that it will provide a list of entries. The first entry is the one that we will be most interested in. How did we know that? Partly because it is the only one with an MIM, or Mendelian Inheritance in Man ID. So let's click that and see what happens. Now we're in the main gene entry for CFTR. There's a lot of information here. First of all, we can see some of the earlier symbols and names for the CFTR gene. We can also see information such as the gene ID number. And let's click down here and go to the reference sequence. Once you have navigated to RefSeq, find the link that begins with the two letters NM and click on that. This will take you to the detailed entry. There's a number of things we can see. First of all, we see that the RefSeq number ends in a version. This indicates the exact sequence that you'll be using. Now let's go to the sequence information. We can see that it is presented in groups of 10 nucleotides. 6 blocks of these 10 nucleotides are presented in each row. Now let's scroll back up to the top. And as you can see, in between there's a lot of information that we'll talk about in future sessions. One important feature in this middle section is the CDS. This indicates that for CFTR, the coding sequence begins at position 133, and we'll cover that in more detail in future sessions. Lastly, let's learn how to save the file. You select Send To, pick File. There are a variety of formats, but the format that we'll choose is the GenBank format. This is a standard that most bioinformatics applications can accept. So in this session, we've learned some basic features of the NCBI resource. We'll go over additional features in future sessions.